slept for 12 hours on Saturday night because I was gosh darn tired. So, all right. Let's take a look at these results real quick. So, actually, Jenny, I gave you back your sheet for this one. So, just to kind of get us going again, why don't you describe to us, I'll have you be the one to report out here. So I think the question is describe the X and Y axis, describe the results or major findings, if I'm not mistaken, or something like that. Okay, so go ahead and get us started. Okay. Okay, so major finding, and I'm going to guess that everybody agrees with this one. There's no big difference between soap with and without triclosan. Was there a difference compared to what was observed at 22 degrees? So did temperature have an effect in hand washing? So take a look at these values here in the gray. These are at around a log of 5 in most cases, at least for those gram positives. S. pyogenes pretty well affected, but between 5 and 6. Again, that means that we're between 100,000 and a million cells per, in this case, milliliter. Okay, let's go back to figure 1. Is there a difference? Not really. Not really, at least not in this context, right? So what's one conclusion we might draw from this? There's no difference. What might we conclude? Yeah, water temperature doesn't have an effect on at least reducing the amount of bacteria on your hands. Does warm water feel better in the dead of winter? Absolutely. I don't disagree with that. Does it make a difference in terms of cleanliness? No, probably not. At least that's what this is arguing. Okay. The one thing to keep in mind, let's just recall real quick, how was this experiment done? So this wasn't done on hands, but this was done in test tubes. Maybe on hands water temperature does make a difference, and that's actually not something that was tested here. And so something for us to consider as maybe a follow-up experiment if we were to follow this up. Hint, hint, you guys have independent studies in the lab soon. This might be a fun one to do. At least that water temperature here doesn't seem to make a difference. Maybe it would on hands itself. Okay? And then mechanical removal of bacteria. All right. Any questions with this figure? Okay, question 6. Who had number 6? Somebody did. I just handed it back. Whitney, what was yours is the diagram, right? For figure 4, right? There's a question that has to do with figure 3. Jill, did I give that one back to you? Okay. Can you read out that question for us real quick? Okay. So let's go ahead to figure 3 real quick. So what's going on here? Okay, perfect. And in this case, what's being measured is just a single bacterium, E. coli. So this is like that previous experiment, but instead of a 20-second exposure, we're observing a long-term exposure here. What are the major, Jill, what's the major difference that we see in this graph?
Well, we just said that the longer the exposure time after being exposed to the soap, the more effective the antibacterial soap was.
Yeah, so I think a lot of people on the responses online pointed out that at 24 hours here, there's a pretty significant difference, at least between soap with triclosan and soap without triclosan. At 72 hours, they're both equally effective. But which one works better faster? Definitely the one with triclosan. So again, this kind of hits on the point of what we talked about. There are two things that are important for physical control and chemical control of growth. What are those two factors? One is concentration, Devin, and exposure time is the other one. Does triclosan work as an antibacterial? Yeah, but how long does it take for us to see those effects? 12 hours, 24 hours, somewhere in that range. Okay? So think of this in terms of when you wash your hands, where does that soap go? In the drain. And where does it go from the drain? Anybody know? Goes to a sewerage district. Basically goes through the sewage system. The concentration that they have here for triclosan, this is controlled in some ways. Right? This is in a test tube exposure to 0.3% over a long period of time. Think about when you wash your hands. I'm going to guess that everybody washes with soap and water. That water is doing what to the concentration of triclosan? Yeah, it's diluting it. So the bacteria, this is in some ways, this is just a proof of principle that, yeah, the triclosan works. But if we think about how this would fit in reality, in the context of everyday lives, bacteria aren't going to be exposed to 0.3% triclosan for a long period of time. In fact, they'll be exposed to much lower concentrations because of that dilution from washing hands. That factor right there, that low concentration exposure, is what's really selecting for bacteria that are resistant to triclosan. And in the same way that we'll talk about on Wednesday, these are the types of factors that lead to antibiotic resistance as well, or selection of microbes that are resistant to low concentrations of an antibiotic. And we'll continue to talk about that over the next few weeks, actually. All right, Whitney, you want to bring your picture up so we can see how figure 4 was done? Okay. That'll work. Okay, go ahead.
Okay, so for ours, we did figure 4, which was they start with hands with bacteriosome, and then it splits into the 3 groups, which is the unwashed, plain soap, and the antibacterial soap. And then they put a glove on and massaged uniformly for 1 minute, and then took the pipette and put it in the glove and then took them off and put it in a test tube with the buffer. And then they plated each 3 of the.
Okay, I love the glove. That's good design. Any questions on this? So basically, again, the bacteria they're using here, which one are they using? Anybody recall? It's colored bacteria, which is really easy to identify. Actually, we'll use it tomorrow in the lab. Allows to tell the difference between, in this case, the bacteria that they've inoculated on individuals' hands versus background contamination. Bless you. So E. coli is kind of a beige color colony. Micrococcus is a yellow. So they wouldn't have counted those. They would only count those that they knew they were inoculating onto the individual's hands. And from this experiment, they get some results. Who answered the question about this figure? I think so, yeah. We're on to 8 now. Is that you, Derek?
Yeah.
All right, so somebody from this group, or Derek, if you want to, take a stab at it.
Well, sorry, so the x-axis was the control, and plain soap, and then wash with antibacterial soap. And then the y-axis was the bacterial preparation, the log CO2. And concentrations, but were more significant than unwashed.
Sure, exactly. Anybody know how to read these box plots? Or did people get generally confused by them? I have to review every single time how to look at these. A couple of key points to bring out here. Basically, those error bars represent your range of 80% of your test population. Okay? So they represent up to the 90th percentile and down to the 10th percentile of those that you've measured. The box represents the middle 50%, between the 25th percent quartile and 75th quartile. And then the solid line is average and the dotted line is median. So there are a couple of ways that folks would probably look at this. I would just take a look at the means and say, is there a difference or not? Some folks might rather take a look at the range, and the reason why they're plotting the data this way is likely because there are going to be some minor differences in hand washing between individuals. And this is trying to account for that variability, okay? This type of measurement where we have these box plots is quite common in experiments with animal subjects, so whether they be human, mouse, chimpanzee, etc., as a way to look at the data. So big take-home point, hand washing works, surprise. Triclosan has no effect on that, on reducing the bacterial population though, right? So what are the major take-home points from this paper then? And this is that last question. The last group I gave a question of, summarize the major points. Meg, what are the major takeaway points from this paper?
We said that the major finding was that the triclosan does not help fight bacterial growth better unless it's been out of person for about 24 hours. There was no true difference in plain soap and antibacterial soap. We should, and another question was if we should ban it or not. And we said that you should ban triclosan because it doesn't help with regular soap and it makes soap more solid and
Yeah, and those reasons right there, the cost of it, the potential side effects, didn't outweigh the potential benefits of it. And that's why the FDA ended up ruling in favor of banning not only triclosan, but I think they banned 19 in total different antimicrobial compounds from hand soaps. And again, as of, I'm not sure if they've come away with a decision from this one, but the only one that they had given an extra year to test were the quaternary ammonium compounds, because those not yet had shown whether they truly were effective in reducing bacterial numbers any better than just regular hand soap. So those are kind of the major takeaway points from here. So the question becomes, what do I want you to be able to do with this? Okay, we've just spent a day and a half talking about this paper. What do I want you to do? I don't want you to memorize the paper. That is not the purpose of this. Purpose, again, is translational information. Can you take what you've learned here in this paper and in our discussion of it and apply it to other data? Let's practice one of those, okay? So I put this one up, and I said that I was going to start doing some of these good, better, best type questions where I would give you a question, and then I'm going to have 3 potential responses where you can self-assess yourself. Do you have a good answer? Do you have an answer that we consider to be better, or is it the best type of answer? Basically a 1, 2, or a 3 on a 3-point scale. So let me zoom in on this, if I can. And here's the question. I want you to analyze the data in this table. This comes from another paper that was published a few years ago, in which they were very similar experiment designed to which was used for Figure 4. But instead of using Serratia marcescens, they took spores of Clostridium difficile and they put it right onto the palm of the hands of their test subjects. And then what they ended up doing was splitting those individuals into 4 different groups. One that received or one that washed hands with Hibiclens, which has chlorhexidine as its base. It's basically I'm trying to remember if it's a phenol or bisphenol. Isogel, Endure, or Purell, which are all alcohol-based sanitizing hand rubs. Okay, so ones that if you work in a hospital, you have probably used something like this. Basically, the goal of those is to wipe it on and to walk out of the room. No washing afterwards. So what I want you to do, they're looking here at the mean reduction in the number of colony-forming units per centimeter squared. So it's basically what's left remaining on the palm of an individual's hand after having washed with one of these different products. What I want you to do, I'm going to give you guys 5 minutes to do this, and I'd like you to actually type or write down an answer for yourself so that you can compare this. And basically answer the question, describe the general trend of the data, so that's part 1, and do that using specific examples from the table. And then next, interpret the results and identify likely explanations. You may not get done with all of this in 5 minutes, and that's okay. But this is the type of translational information that I want you to be able to do. Similar question that we've seen before. Okay, so first question. And use your tables for this if you want to. Okay, this is meant to be a practice. Which of the following antibiotics directly inhibits translation? Is it a beta-lactam, a quinolone, bacitracin, or the aminoglycosides? Should we actually check how we're doing? Yeah? Maybe? Okay. Looks like we're doing pretty good. Aminoglycosides. We'll come back to these other ones. What others directly inhibit translation? So what of the other ones that I asked you about directly inhibit translation? Devin. Rifampin? Rifampin actually will target RNA transcription. So yes, there's going to be an effect on translation, but it's not directly inhibiting the process of translation. Okay? We'll come back to that one. There's at least one other one, I think, on that list. Brendan? What's that? The chloramphenicol? Yeah, that one definitely will. No, go ahead. Go ahead.
No, that's it.
Okay. Yeah, so that one will work as well. So the aminoglycosides, there's another one that's not on the list, which are the tetracyclines that also inhibit translation. When we say that these drugs inhibit translation, what specifically are they binding to inhibit that translation? Yeah, the ribosome. So, and this is why I had you guys do that practice with those concept maps on for Friday, was to just remind yourself things like what's involved in translation. It's the ribosome. Most of these drugs that inhibit translation all bind to either the large or the small subunit of the ribosome, and in some way inhibit its ability to read the mRNA, and then bring those have those tRNAs bring amino acids and produce a polypeptide. Okay. All right. Next one. Which of the following is not part of triple antibiotic ointment? This is actually not on your list. But some of these are actually on your list. So is it neomycin, tetracycline, bacitracin, or polymyxin B? The other name for triple antibiotic ointment, Neosporin, might give away the answer on one of these that is involved. Who thinks they know the answer? Who's confident? Austin? Yeah, tetracycline's not involved. The other three are. So we just said that tetracycline inhibits translation. What about these other ones? What are the targets of neomycin? So what does neomycin block? Protein synthesis. So it inhibits the ribosome specifically. What about bacitracin? Was this one on the list? Okay. Yeah, cell wall synthesis would be the way that I would describe this one. Narrow spectrum. What do we mean by narrow spectrum drugs when we are talking about antibiotics? Kaylin? Yeah, essentially it targets only a small range of bacteria. And so bacitracin, because it targets the cell wall, what does it work against? It's a narrow spectrum drug that works against which types of microorganisms? Oh, we got a mix here. Okay. What are a couple of differences between gram positives and gram negatives? Thickness of the wall is one. What else? Outer membrane on gram negatives. Bacitracin can't pass through that outer membrane. And so in fact, bacitracin is selective for gram positives. In other words, it can inhibit, Abby, are you questioning what I'm saying?
So but said it can do some gram negatives.
It may be able to do some, but in general, I talk about them as being specific to gram positives. Maybe it's a reductionist approach that's oversimplifying